Hello, everyone. Welcome to the webinar, Advancing DILI Assessment Using Micropatterned Co-Culture Models, HEPATOPAC and HEPATOMUNE. This is the second in a series of 5 webinars in which we'll be talking about novel applications for HEPATOPAC. I'm Andrea Wolf, and I'll be facilitating the webinar today. I'm a director at BioIVT's lab in Kansas City, the legacy Xenotech site, where we offer a comprehensive portfolio of in vitro ADME services and products. Before I introduce Sarah, our speaker on today's webinar, I'd like to take a few minutes to tell you about BioIVT. Founded in 1981, BioIVT has grown to become the largest provider of biospecimens and research services to the pharmaceutical and diagnostics industry. We are dedicated to providing the biospecimens and services that our customers need to help them advance their discovery in preclinical and drug development programs. We have grown both organically and through strategic acquisitions that have enabled us to develop a comprehensive product portfolio and become the preferred provider for our global biopharmaceutical clients. As a legacy Xenotech scientist, I want to highlight that Xenotech joined the BioIVT family in 2022. This significantly expanded our research services and product capabilities. As a director at our Kansas City facility, I have over 25 years of experience working with our clients to design and implement in vitro drug transporter studies to assist them in achieving their research objectives. One of our unique strengths is that we can partner with our clients to design studies using both conventional and custom methods. When conventional methodologies might not be sufficient to answer our clients' questions, we can employ other technologies and customizations to answer your specific research questions. For example, we can utilize the long-term HEPATOPAC model in our studies to evaluate low turnover and slowly transported compounds. In relation to our product portfolio, we have made strategic investments in sourcing biospecimens globally and an extensive global distribution. We stage biospecimen products in regions where our customers are located, and this helps us to ensure short turnaround times, enabling us to get products to our customers' labs when they need them and keep their research programs on track. At the foundation of everything we do is a steadfast commitment to ethically sourcing, uncompromising quality, and a genuine dedication to partnering with the research community. Here is a snapshot of the array of BioIVT's portfolio, including products used in cell and gene therapy programs, normal and disease state biospecimens, and products and services to evaluate the ADME property of drugs. Our ADME group has the industry's largest portfolio of hepatocyte subcellular fractions and recombinant enzymes, as well as other products to support ADME tox and DMPK research. Our team of experts routinely works with clients to help them identify hepatocyte lots, having the characteristics needed to meet their specific requirements. On the ADME services side, in addition to the transporter studies I mentioned earlier, we work with our clients to design and conduct IND-enabling packages. These include studies such as metabolism, enzyme induction and inhibition, as well as other mechanistic studies to predict the pharmacokinetics of drug candidates. Our ADME Services Group provides individual studies as well as integrated programs based on our clients' needs and challenges. Many of our clients start with a consulting engagement in which we provide expert advice to design ADME programs. Then we can also assist with gap analyses and analyze existing data packages or help prepare for regulatory meetings. We offer full in vitro ADME programs at our labs in Kansas City, Santa Clara, and Durham. When clients choose to work with us, they get the benefit of a team of experts who collectively have worked on thousands of programs and bring that experience to each new project. If you're interested in or have questions concerning drug transporter studies or any of our service offerings, I'd be happy to talk with you. My contact information as well as Sarah's are provided at the end of this webinar. Additionally, we have partnerships that enable us to integrate in vivo studies into preclinical programs, and we have extensive capabilities for bioanalysis to support these programs or to offer as a standalone service. It is my pleasure to introduce Sarah Garish. Sarah is a PhD student in pharmaceutical sciences at the University of Maryland, Baltimore. She works with Dr. Hong Bing Wang in his lab, and she holds a bachelor's degree in clinical pharmacy and a master's degree in pharmacology from Georgetown University. Her research focuses on drug-induced liver injury and metabolism-based drug-drug interactions using advanced human relevant liver models. Her current work integrates functional assays and transcriptomic profiling to uncover hepatotoxicity mechanisms and biomarkers, contributing to FDA-funded efforts aimed at improving preclinical drug safety evaluation. Sarah, we're very much appreciative of you joining this webinar series on HEPATOPAC and sharing your results from your research work with us today.
Thank you so much for inviting me, and welcome everybody. Thank you for joining the webinar, and thank you for the introduction as well, Andrea. So today I'll be presenting our work on drug-induced liver injury using HEPATOPAC and HEPATOMUNE models. And the title of my presentation is Advancing DILI Assessment Using Micropatterned Co-culture Models, HEPATOPAC and HEPATOMUNE. So I first want to give a brief background on drug-induced liver injury, or DILI for short. DILI is a serious side effect that could happen to many medications affecting the liver, causing liver damage. So it can happen to many of the drugs, including over-the-counter medications and also herbal supplements. And the severity of DILI can range from mild liver enzyme elevation, such as ALT, AST, to severe liver failure, and sometimes can cause death. So first, I want to start by why is it important to study DILI, or what is the significance of DILI, or drug-induced liver injury? So although the disease is rare, it could have some severe consequences, especially since DILI has been the main cause for acute liver injury in Europe and the US. And also it has been a main reason for post-marketing drug withdrawal and clinical trial terminations, which all, of course, affect patient safety. And another problem is the financial burden of drug development, especially when it reaches the clinical trial phase. And next, I just want to go into the different classification of DILI. So DILI can be classified into two main types, intrinsic and idiosyncratic. And intrinsic DILI is dose-dependent and drug-dependent, which it depends more on the intrinsic characteristics of the drug. And it has a direct hepatotoxicity effect. So that makes it easier to be predicted in vitro models or even animal models, because the pattern of toxicity is much easier to predict when that compared to idiosyncratic DILI. On the opposite side is idiosyncratic DILI, and for idiosyncratic DILI, it's dose independent and also drug independent, but it depends more on the host factors. It depends more on patient susceptibility, so it could happen in some patients and not in the others. And it's characterized by a complex disease pathogenesis, which makes it more challenging to predict compared to idiosyncratic DILI. And here's the blue arrow is indicating that by increasing our understanding of the mechanism involved in drug-induced liver injury, specifically IDILI or idiosyncratic DILI, that will help us to better predict hepatotoxicity before the drug reaches the patient. So I just included this slide to show how many different mechanisms can be involved in drug-induced liver injury. And they can be classified into immune-mediated and non-immune-mediated, starting from mitochondrial dysfunction, ROS formation, also and transporter inhibition, lysosomal impairment. So this just shows that there are many mechanisms involved, specifically for idiosyncratic DILI drugs. And one drug could have different mechanisms of hepatotoxicity. And also for DILI testing across the drug development pipeline, the drug passes through many stages, starting from in vitro models, in vivo models, and finally clinical testing in human clinical trials. And the problem here with DILI drugs is that the drug can pass through all these stages and still cannot be detected for its hepatotoxicity. And that's why sometimes it gets withdrawn from the market or even at the clinical trial phase. So this highlights that there are some problems with the conventional methods in which we like in DILI detection. And I'll highlight some of the problems in vivo models, for example, for animal. So one of the main problems in using animal models is that species-specific differences, especially in drug metabolizing enzymes, and also which makes it harder to predict DILI sometimes. Like for some of the drugs, they won't show toxicity even at supratherapeutic doses. And also another problem is that sometimes they show different patterns of toxicity, which affect their translational value. And next I'll move into in vitro models, which is the main focus of today's webinar. And in vitro models, or liver in vitro models, involve many cell sources, starting from primary hepatocytes. Also, several cell lines are available. And there are many cell types, either parenchymal or hepatocytes versus non-parenchymal cells, which all can be involved in hepatotoxicity. And finally, it could be in different configurations, either 2D or 3D configurations. And there are pros and cons to each of them. So if we move from the 2D configuration to 3D configuration, there is an increase in physiological relevance, and also having co-cultures or different types of cells facilitates or allow cell interactions, which can help us see things that we can't see in regular monocultures. However, there are also some difficulties as we increase the complexity. It could be a little bit difficult in handling, or sometimes it lowers the throughput as well. And primary human hepatocytes has been the gold standard in metabolic studies and toxicity studies for a long time. And this is because they can retain the functionality of the liver. However, there's a downside in using primary hepatocytes in 2D conventional culture, is that they rapidly decline in function, which makes it harder to test drugs that could be causing long-term toxicity, or that needs long time to show effect. However, by the use of novel bioengineering tools, this has enabled stable hepatocyte culture, or for a longer period of time. And this is where I introduce the micropatterned co-cultures, HEPATOPAC and HEPATOMUNE, and I'll have more details on the next slide. So I'll start first by HEPATOPAC. HEPATOPAC is a micropatterned co-culture system, and it's created by microfabrication tools, where collagen gets first fixed on islands, like the circular shapes here, and then hepatocytes attached on collagen islands. And finally, murine fibroblasts will be seated and surrounding the islands of hepatocytes. Here is another magnified image of a hepatocyte island showing biochemical eye. Next is HEPATOMUNE, and HEPATOMUNE is pretty much similar to HEPATOPAC. However, there's just the addition of Kupffer cells, inclusion of liver macrophages, Kupffer cells, which helps to make them more immune competent or help to make them respond to inflammatory stimuli. And here I have two images comparing HEPATOPAC and HEPATOMUNE, where in HEPATOMUNE has green fluorescence due to CD68 staining of Kupffer cells. So this helped to test the immune component of drug-induced liver injury, for example. Then I want to highlight why we choose to use these models. There are many advantages to the HEPATOPAC and HEPATOMUNE cultures, starting that they are relatively easy to handle compared to other more complex systems. And also having co-culture or different types of cells from hepatocytes to fibroblast and finally Kupffer cells makes it possible to observe cell-cell interactions. And also I'm going to show some data about the functionality of these models. So this is albumin and urea production. So this shows that the cells remain viable and able to produce albumin and urea for a long period of time, up to 28 days. So here I'm comparing pure hepatocytes in the closed circles, and open circles are HEPATOPAC model. So this shows consistent albumin secretion, and similarly in urea, where also the levels are much higher when compared to the decline in the pure hepatocytes culture. Next, they retain the activity of phase 1 and phase 2 enzymes for also a longer period of time, up to 20 days, as shown here in the gray bars, compared to pure hepatocytes culture of just 11 days, as shown by the metabolism of, these are the metabolites of the test drugs. And also they express liver metabolizing enzymes, also phase 1 and phase 2, for a longer period of time compared to hepatocytes. So this all shows that they are metabolically competent. And finally, for HEPATOMUNE, as it includes functional Kupffer cells, this can help to detect immune-related DILI or toxicities. And here, this data shows that some cytokine release in response to LPS, which shows the activity of Kupffer cells. And on the other side, this is like a phagocytic response to Staph aureus after 10 days of Kupffer cell culture. So this brings up our research question, which is, can advanced in vitro MPCC models such as HEPATOPAC and HEPATOMUNE improve the prediction of drug-induced liver injury, and at the same time help us to understand the mechanism behind the toxicity? And to test cytotoxicity in these models, we had to select a list of drugs, and with the help of our FDA collaborators, we came up with 23 drug pairs, and each pair needed to be structurally and mechanistically similar, which they have the same mechanism of action, but they needed to have different toxicity profile, in which one drug is toxic and the other one either less toxic or non-toxic compared to the other one. And so the idea behind the criteria of choosing these drugs was to make them comparable as much as possible and exclude other obvious factors that can be contributing to DILI. We want to understand why drugs from the same family, and one of them is causing DILI and the other one is not. So this consisted of a 3-tier approach, and the first tier included DILI risk factors such as daily dose, DOCP, and reactive metabolites. So the drugs needed to be comparable, in which either the two drugs form reactive metabolites or not. And also for the daily dose, it shouldn't be, the toxic drug shouldn't be 10-fold higher than the non-toxic one. And also for log-P or lipophilicity, the difference should not exceed 1.5 units. And the other tiers excluded drugs that were metabolized by different interspecies differences in CYPs and different animal toxicities. And finally, the commercial availability of these drugs were considered as well. So we came up with an initial list of pairs for the testing in these models, which are trovafloxacin and moxifloxacin, dabigatran, ibiphenec, and ibuprofen. But for the sake of this presentation, I'll be mainly focusing on the first one, trovafloxacin and moxifloxacin. So this is a comparison between trovafloxacin and moxifloxacin, showing their structures and the similarities. They are both fluoroquinolone antibiotics, which have the same scaffold, quinolone core, and similar chlorine substitutions, carboxylic acid, and so on. And they just differ in some of the substitutions here, which were shown in some studies to be connected to or linked to hepatotoxicity. And they have the same mechanism of action, but one of them is a DILI, most DILI concern, or the DILI-toxic drug, which was withdrawn from the market, which is trovafloxacin. And the other one is moxifloxacin, which is relatively safer than trovafloxacin and still marketed with some like rare hepatotoxic events. So for the timeline, the process starts by seeding hepatocytes on day 0, and then fibroblasts will be seeded on the next day or after hepatocytes attach. And there's a stabilization period of 7 days, during which Kupffer cells will be seeded in hepatomune from day 5 to day 7. And then the plates are checked on the 7th day or after 1 week. And after we receive the plates, we change the media and keep it for a 2-day period recovery. And then we start our treatment with trovafloxacin and moxifloxacin at different range of concentrations, depending on their Cmax. And we also included another model in which it had hepatomune plus LPS as an inflammatory stimulant. And we just exchanged the medium every other day, and finally measured cell viability by measuring ATP. So this is the cell viability results of trovafloxacin and moxifloxacin. This is those response curves showing that trovafloxacin is the toxic drug versus moxifloxacin, which is not toxic. And also the IC50 of the hepatomune plus LPS model was the lowest, indicating that LPS has sensitized hepatomune and made it more susceptible to trovafloxacin hepatotoxicity. Next, we also wanted to see cytotoxicity under the microscope using live blood staining. So we used trovafloxacin at a dose that is at the IC50 dose, 125 micromolars, and the corresponding dose of moxifloxacin. And we also included the control. This was done in a pooled HEPATOPAC model consisting of different donors. And it was also evident that trovafloxacin is the toxic drug, where it has less viable cells and more dead cells by PI staining. Then, based on that literature, trovafloxacin has been shown to cause toxicity in several mechanisms, which also reflects its idiosyncratic nature, or idiosyncratic drug-induced liver injury nature. And these mechanisms involve bioactivation of active metabolites to mitochondrial dysfunction, also RS formation, and also it has immune-related hepatotoxicities, and finally, disruption of cellular signaling and apoptosis. So we wanted to look into these different mechanisms further, but before I move into the apoptosis results, I just want to show a summary of some in vitro studies that were used to understand the mechanisms of trovafloxacin toxicity. So most of these studies include the co-inflammatory stress or co-inflammatory factors such as low-dose LPS or TNF-alpha, and sometimes other cytokines as well. And for example, for this study in which they were testing or trying to understand the mechanism behind apoptosis, they also included TNF-alpha in their study. So we think it would be great if we can see the same endpoint without having another external factor such as LPS or TNF-alpha. And for that, we here tested caspase 3/7 activation or measured apoptosis. And this was done in a pooled hepatomune model of different donors. And I have trovafloxacin in red, and the darker the red color, the higher the concentration, versus moxifloxacin in green. And it was showing that by increasing the concentration of trovafloxacin, there was a greater activation of caspase 3-7, and it was also increasing by time. So this shows like a dose and time-dependent increase in apoptosis by trovafloxacin treatment. So next, we also looked into the immune-related inflammation. And here we used celsupernatant at a dose that is 25 Cmax or 100 micromolar, which is also close to the IC50. And we used a Luminex assay to measure a bunch of different cytokines, including interleukin-1 beta, interleukin-6, interleukin-10, and finally TNF-alpha and interleukin-8. So the main thing I want to highlight out of these bar graphs is that for most of the other cytokines, it wasn't showing elevation or increase in the protein level unless LPS was added. However, that wasn't the case for interleukin-8, in which we were seeing an increase in the protein levels, even without the inclusion of LPS. So, of course, with the inclusion of LPS, the level was even higher, but we were still seeing some increase in the HEPATOPAC and HEPATOMUNE models, even without LPS. HEPATOMUNE plus LPS is the green bar versus HEPATOMUNE blue, and finally HEPATOPAC is in purple. So this is what first brought our interest in interleukin-8. And another interesting finding was that HEPATOPAC by itself was still able to secrete interleukin-8, which shows like just hepatocytes can also secrete cytokines. And more into interleukin-8. So interleukin-8 is a pro-inflammatory cytokine chemokine, which is responsible for recruiting neutrophils and other immune cells. And it has been implicated in some studies where it contributes to liver damage and may exacerbate inflammation and injury. So this is the same result from the previous assay, but with full change analysis. In day 5, comparing HEPATOPAC versus HEPATOMUNE IL-8 secretion in response to trovafloxacin. And this shows that IL-8 level was significantly increased in HEPATOMUNE compared to HEPATOPAC versus moxifloxacin, which wasn't shown as like a big change or increase in IL-8 level. So going back to our research question, so to the next half of the research question, which is the mechanism. So far we have been looking at specific endpoints, whether cytokines or cytotoxicity. But then we wanted to see a broader view of hepatotoxicity, in which we did a transcriptomic study in order to see this different dysregulated genes or pathways involved in trovafloxacin hepatotoxicity. And the purpose behind this was to try to understand more about the mechanism of toxicity and also identify early biomarkers of hepatotoxicity. I'll go briefly into the transcriptomic workflow. We used pooled HEPATOPAC and HEPATOMUNE model to account for inter-donor variability. And we used the same concentration of trovafloxacin and moxifloxacin because we wanted to see drug effect rather than concentration effect. At IC20 dose of trovafloxacin, because we also wanted to see early toxicity, or like we wanted to identify early biomarkers before the onset of severe toxicity. So that's why we used a lower dose rather than IC50. And then RNA was extracted and sequenced, and finally analysis was done to identify DILI biomarkers. I just want to highlight one step here in the transcriptomic analysis, which is this one. So since we used, since there's murine cells, murine fibroblast cells included in the culture, we wanted to exclude the mouse genes. So we just added another step to the analysis where we just extracted all the mouse gene to only have human genome in the end. Like we removed all the mouse genome, and then expression analysis was done to identify differentially expressed genes and also dysregulated pathways. So here I'm showing that trovafloxacin have induced much greater changes compared to moxifloxacin on the gene expression level, either for upregulated genes or downregulated genes. Also, comparing HEPATOPAC to HEPATOMUNE, there are much bigger change in the trovafloxacin group compared to moxifloxacin. And here I'm also showing heat maps of different genes, and this shows like how different the expression profiles of the two drug groups are, where different genes are being differentially expressed. Same case in HEPATOPAC and HEPATOMUNE 1. And usually trovafloxacin will also have a higher fold change. In this slide, I'm showing the top 20 significantly upregulated pathways upon trovafloxacin treatment. And the size of the dot reflects the number of genes, and the more the red color is, the more the significance. So here I'm comparing HEPATOPAC to HEPATOMUNE of upregulated genes in response to trovafloxacin. And it was shown that trovafloxacin activates the stress and inflammatory related pathways, and also pathways that can cause cell death. Also, we see a more cytokine related pathways and immune related pathways, and the immune context of HEPATOMUNE is a little bit higher than when compared to HEPATOPAC, especially since inclusion of Kupffer cells. Also, for the PI3K ACT signaling pathway, it was shown to be involved in interleukin-8 production in some of the studies, which also can be interesting for our study since we're interested in interleukin-8. Next on this slide, I have also the top significant downregulated genes comparing HEPATOPAC and HEPATOMUNE, and it was shown that trovafloxacin affects pathways that are related to hepatocyte function and metabolism. So the metabolism of drug metabolism and also CYPs, bile acid secretion and synthesis, and also for downregulation of PPAR signaling, which is important for fatty acid metabolism and other liver metabolism. And some of the pathways can be overlapped, in which they could have some overlapping genes. And also for some of the other pathways here involving sugar, amino acid, and fatty acid metabolism were affected. And this can be related to the mitochondrial dysfunction aspect of trovafloxacin toxicity. So they all these pathways correlate with the identified mechanisms of trovafloxacin hepatotoxicity. And coming back to interleukin-8, it was also shown to be its expression was also elevated in our mRNA sequencing results. So here I'm showing some volcano plots where the blue are the downregulated genes, red are the upregulated genes. And as we move further from the center, that would signify increase in significance. So interleukin-8 was seen to be upregulated in the HEPATOPAC and HEPATOMUNE models, with a further increase in the fold change in the HEPATOMUNE model. Here I'm showing normalized mRNA expression results in HEPATOPAC versus HEPATOMUNE at the IC20 dose of trovafloxacin. And also, I just included this slide to show that trovafloxacin was enriched in top significant pathways, top significant upregulated pathways in both HEPATOPAC and HEPATOMUNE. So the mRNA expression of interleukin-8 was elevated in our RNA-6 studies. Also, the protein level was elevated, and finally, it's involved in top enriched pathways. So this all support that interleukin-8 can be a potential candidate for trovafloxacin toxicity, warranting further studies or additional studies to see if interleukin-8 is actually contributing to trovafloxacin hepatotoxicity. And for that, I've included some next steps in testing interleukin-8, where we might use anti-interleukin-8 antibody to block its activity, or also small molecule inhibitors to block interleukin-8 receptors. And finally, we can use interleukin-8 siRNA to knock down its gene expression, and also assess phenotypic effects at the end. So from my conclusions, HEPATOPAC and HEPATOMUNE, both of them showed trovafloxacin hepatotoxicity with an added immune function in the HEPATOMUNE model. Also, trovafloxacin caused time and dose-dependent apoptosis or activation of caspase-3/7. Finally, trovafloxacin also caused some greater changes in the gene expression of HEPATOPAC and HEPATOMUNE compared to moxifloxacin by itself. And some of the stress and inflammatory pathways were activated by trovafloxacin treatment, opposite to some of the pathways involved in metabolism and function of hepatocytes that were downregulated. And finally, interleukin-8 was consistently upregulated in the mRNA and protein levels. So this highlights that interleukin-8 can be a potential candidate for further study related to trovafloxacin-induced hepatotoxicity. Finally, I want to thank my lab. I want to thank Dr. Wong, Lin Hao, and Kiwi, and also Ritika, which was a previous member of our lab. So I want to thank my IVT team, and our FDA collaborators, and everyone. Thank you so much for contributing to this work. It wouldn't have been possible without the help of everybody. Thanks. And with that, I'm happy to receive any questions.
I also just want to share that we have quite a few resources available on our website for HEPATOPAC. And if you visit the resource page and search for HEPATOPAC under the educational content, you can find training videos, user guides, brochures, and publications for HEPATOPAC in case you're interested in purchasing this product, or also using it as or maybe doing a service with us where we would do it for you. So, and just as a reminder, this is the second in a series of webinars, and if you missed the first one, it's now available on demand on our website, so you can go there to download it. And if you haven't already, please register for the next webinar on October 9th, where John Griffith will share his innovative work on siRNA delivery in HEPATOPAC. Thank you very much for your attendance today. And as mentioned earlier, here's my email address as well as Sarah's. And you're welcome to contact us directly if you have any questions later after you've had a chance to think about the work that she's done. You can also contact us via the website at the address shown here. You'll receive a short survey to provide your feedback on this webinar and how we can better serve you in the future. So thank you again for your attendance today.
Thanks so much, Andrea. Thank you.
Thanks, Sarah.